Every living thing is composed of cells. Your body is made up of trillions of cells, yet at conception, you are no more than a single cell formed from the union of a sperm and an egg. All cells come from other cells. From that single cell at conception, you developed into your current size and proportions through repeated cell divisions. Even now, your cells are continually dividing to replace dead or damaged cells. In single-celled organisms, such as diatoms, cell divisions bring about new individuals. In multicellular organisms, such as yourself, cell divisions result in growth or replacement of cells. There are two major types of cells, prokaryotic and eukaryotic. Prokaryotic cells do not contain a membrane-enclosed nucleus. DNA, the cell's genetic material, is located in an area of the cell called the nucleoid region. There are no membranes to separate the nucleoid region and the DNA from the rest of the cell. Bacteria and cyanobacteria are prokaryotes. In contrast, the eukaryotic cell has a nucleus that is bound by a double membrane, or nuclear envelope. DNA is located within the nucleus. The entire area outside the nucleus and within the cell membrane is called the cytoplasm. Membrane-bound organelles with specialized functions and structures reside within the cytoplasm. Eukaryotic cells are present in cysts, plants, fungi, and animals. This is a picture of some eukaryotic cells. The structure in the center of each cell is the nucleus. The cytoplasm is the watery substance surrounding the nucleus. Within the nucleus of a eukaryotic cell, DNA, the cell's hereditary material, is compacted along with proteins into thread-like structures called chromosomes. DNA is very important to the cell. Within it lie genes, which carry the cell's hereditary information. The genes give directions for everything the cell is and will be. Although prokaryotic cells also contain DNA, it is not enclosed by a nucleus as it is in eukaryotic cells. Usually in prokaryotes, the DNA makes up a single circular chromosome. This is an electron microscope image of a circular chromosome isolated from a bacterium. The method of cellular reproduction differs between prokaryotic cells and eukaryotic cells. Prokaryotic cells reproduce by a type of cell division called binary fission. In binary fission of bacteria, the chromosome replicates. The cell grows about double in size and then divides the parent cell in half to form two daughter cells, each with the same DNA as the original cell. Eukaryotic cells divide by mitosis, which is division of the nucleus, and cytokinesis, division of the cytoplasm. Before a eukaryotic cell can divide in half, its nucleus must be duplicated so that both the parent cell and the new cell will receive an identical set of chromosomes. A eukaryotic cell goes through a distinctive cell cycle. This diagram represents the different phases of this cycle. The G1, S, and G2 phases make up interphase. During interphase, the cell grows, producing more cell structures and doubles its chromosome set. During the G1 phase of interphase, the cell grows and more proteins, nucleic acids, and cellular organelles are produced. During the S phase, each chromosome duplicates to form another identical chromosome. These remain attached together by a centromere. The individual members of the attached pairs are called chromatids. In the last phase of interphase, G2, the cell continues to grow and metabolize normally and readies itself for the M phase. Mitosis, the first part of the M phase, consists of 4 stages: prophase, metaphase, anaphase, and telophase, during which the cell's nucleus divides in half. Mitosis is followed by cytokinesis. during which the cytoplasm of the cell is separated into two equal portions. Of the four different stages of mitosis, the first stage, prophase, is the longest. When cells enter prophase, their chromosomes are doubled, elongated, and indistinguishable. During the first part of prophase, the chromosomes condense and become visible. In animal cells, two pairs of rod-shaped structures in the cytoplasm, called centrioles, begin to separate and move to opposite sides of the cell. Initially, in prophase, the nucleus is very prominent and contains a distinguishable nucleolus. As this stage progresses, however, the nucleolus and nuclear envelope enclosing the nucleus disappear. From the centriole pairs, fiber-like microtubules extend to form a spindle. Some of the microtubules in the spindle attach themselves to each chromatid of the chromosomes. With the aid of the microtubules, the chromosomes move toward the equator of the cell, called the metaphase plate. This is a picture of animal cells at the end of prophase. Notice how the nucleus and nucleolus are no longer distinct, and the chromosomes are very condensed. During the second stage of mitosis, metaphase, the chromosomes line up along the metaphase plate. These pictures show plant and animal cells undergoing metaphase. Plant cells lack centrioles, but microtubules enable the chromosomes to align at the metaphase plate. During anaphase, the third stage of mitosis, the sister chromatids of each chromosome separate and move toward opposite poles of the cell. This happens in such a way that each side of the cell receives one complete set of chromosomes. The plant cell shown in this picture is a clear example of a cell during anaphase. Note the sets of chromosomes at opposite poles of the cell. Anaphase is followed by the final stage of mitosis, telophase. The chromosomes continue to move toward the poles of the cell. The spindle then disappears. Nuclear envelopes form around the chromosomes, and nucleoli reappear in the newly formed nuclei. Mitosis, the division of the nucleus of a cell into two identical nuclei, is now complete. By this time, cytokinesis is usually well underway. This picture shows a plant cell in telophase. Notice the two distinct nuclei. Cytokinesis, unlike mitosis, differs a lot between plant and animal cells. In animal cells, the membrane surrounding the entire cell pinches in at the middle, dividing the cytoplasm in half. In plant cells, however, the cytoplasm is divided by a cell plate. From this cell plate, two new cell walls form, one for each of the two plant cells. This picture shows the result of mitosis and cytokinesis in animal and plant cells. The newly formed cells are referred to as daughter cells. It should now be clear that mitosis and cytokinesis provide a way to form two eukaryotic cells from one. Repeated cell divisions allow multicellular organisms to grow and develop and replace dead or damaged cells. While studies suggest that something in the cell's cytoplasm triggers cell division, the exact initiating factor remains unknown. This is one of many mysteries in cell biology that keep scientists working busily.